Hi, Michelle Glass here. Welcome back to our second installment in our Chapter 2, Part 2. So we're actually in our lecture 2.10. And our topic for this video are those carbohydrates. So we're looking at our first category of organic macromolecules, or we can also call them organic biomolecules, where bio, of course, means life. So that term biomolecule would be molecules of life. Now, as we look at our carbohydrates, their primary job is to be our primary source of energy. So about 60% of the time, our cells are using carbohydrates in order to make energy. We see then that this is the ideal for our cells. When you're eating food, you're eating carbohydrates, so you're going to be flooding your bloodstream with glucose, and so your cells are going to be taking that glucose and using it to make energy. So when we talk about carbohydrates as our primary source of energy for humans, our cells prefer glucose. So we can consider glucose to be the primary monomer or building block as we're talking about carbohydrates in our body. So all of the carbohydrates that we eat will be broken down to the monomer glucose. In our cells, if we have extra glucose, our liver cells specifically can store that glucose in the form of glycogen. So glycogen would be an example of a polymer where you have lots of glucose molecules chained together. So think of glucose as the monomer that our cells are using to make energy. Glycogen is our polymer that our liver cells and some of our skeletal muscle cells will use to store glucose. As we talk about carbohydrate structure, we will see that carbohydrates are composed of carbon and hydrogen and oxygen, and we get a ratio of 1 carbon for every 2 hydrogens and then 1 oxygen. So it's a 1 to 2 to 1 ratio, carbon, hydrogen, and oxygen. Now, as you're looking at your structure here, you can see that carbohydrates can either be listed out or drawn in a linear structure or in a ring structure. The ring is the more typical shape that you'll see for your carbohydrates. And all we've done here is use our points to represent our carbon, just to kind of simplify our drawing. So notice that we have hydroxyl groups, that's that OH group there, and here we have the carboxyl group, I don't think I said the right name, hydroxyl group here as well. So we see that throughout. It's going to actually make our carbohydrates hydrophilic, meaning they love water. So you already know that sugar will dissolve in water. So you can use that as a memory device. Now, that basic structure, that ring that we were looking at, where you have the ratio of 1 carbon, 2 hydrogens, 1 oxygen, really is representing our monomer. And we talked about glucose as being our monomer, but there are lots of different monomers in our carbohydrates. So really, our best description of the monomer for carbohydrate is a monosaccharide, where mono means 1 and saccharide means sugar. So you can also call these single sugars or simple sugars. Glucose is what we're going to primarily use in our bodies. Fructose is another example, and this is something we eat all the time. Fructose is fruit sugar, so that's what makes those grapes and apples and oranges super tasty. When we eat that fructose, then our body will break it down into glucose or convert it to glucose for our cells to use. When you link together 2 of those monomers, you get what's called a disaccharide. So di means 2 sugars. And a good example of a disaccharide is sucrose. Sucrose is our table sugar, so we're very familiar with that one as well. Notice that glucose and fructose and sucrose all have the same ending. They all have an OSE ending. So that's going to be indicative of carbohydrates. What that means is if you see the name of a molecule that ends in OSE, you should assume that it's a carbohydrate. But not every single carbohydrate has that ending. So we've already seen glycogen, which does not have that ending, but it is still a carbohydrate. So how would you actually combine monosaccharides into a disaccharide. Let's take a look at that reaction. Oh, well, I jumped ahead. So let me jump ahead and come back, okay? So I liked my transition. So here we go. This is our picture of how we're going to take two monomers. So here's glucose and fructose, and then perform dehydration synthesis in order to make sucrose, which is our disaccharide here. Remember, dehydration means to remove water, so we're going to pull out a hydroxyl group and a hydrogen here, and that's going to form water. H2O is, of course, water. And so we still need to complete the bond with oxygen, and so that's going to be the bond that's linking together these two monosaccharides into a disaccharide. When you're eating sucrose, then you're going to do the reverse of this reaction. So that's what we're looking at here. So this is the reverse. So now we've written it from still left to right. So we're starting with our sucrose, our table salt. Our cells are using water, breaking that water molecule down into a hydroxyl and a hydrogen, breaking that bond between the glucose and the fructose. So the hydrolysis reaction, remember, is a catabolic reaction or breakdown. The dehydration synthesis is an anabolic or making reaction. Going back to this slide here, so we've mentioned monosaccharides as our monomer. We've said you can link them together to create a disaccharide. When we talk about glycogen, is actually what's called a polysaccharide, where poly means many. So that's lots and lots of sugar molecules, simple sugar molecules attached to each other. We can take a look at our glycogen molecule here. So notice all of these glucose molecules. Glucose, remember, is your monosaccharide. It's our source of energy. So in our cells, we're going to break down glycogen in order to release glucose, in order to make our energy molecule, which we're going to be naming here shortly. It's ATP. And then in both our liver and our skeletal muscle, we do have the ability to store glucose in the form of glycogen. So this is how our bodies will store any extra glucose molecules that we might get from our diet. And that is it for now. Stay tuned.